Hello, Assalamualaikum. Welcome back to my video. So during previous video, we already learned number 1 and number 2. Now we move to the detailed steps in mitosis. As I said earlier, mitosis consists of 4 subphases: prophase, metaphase, anaphase, and telophase. We will look at the subphases one by one. So now we start with the prophase. So prophase, look at the diagram. The nuclear envelope starting to disappear, and then the chromatin slowly to condense, whereby it tightly coil and it becomes shortened and thickened. So the chromosome now consists of 2 sister chromatids, whereby it attached by the centromere, and then the mitotic spindle, aka the microtubule, will grow up from a centrosome, each chromosome, both chromosome, and it migrate to the opposite poles of cells. The centrosome migrate to the opposite poles of cell. During that prophase, the mitotic spindle attach the kinetochores of the each chromosome. Okay, next step is the prometaphase. Even though prometaphase is not in the major subphases in mitosis, but it's still including, it is the addition of the step in mitosis. Prometaphase before metaphase. So in prometaphase, the nuclear envelope fragmented. You can see that the nuclear envelope of the cell is nuclear envelope of the nucleus already disappear, and then each chromatid of chromosome has kinetochore. The kinetochore denoted by the black piece, okay, black piece, and then some of the microtubules attached to the kinetochore. So the microtubule that attached to the kinetochore, we call it as kinetochore microtubule, and then the non-kinetochore microtubule, we can see that they interact with those from opposite poles of the spindle, and then all chromosome move toward to the metaphase plate. So metaphase plate meaning the middle of the cell. Alright, so now it comes to the metaphase. Next step is the chromosomes. All chromosomes line up along the metaphase plate. You can see that all chromosomes are in the middle of the cell. For each chromosome, the kinetochores of sister chromatid attached to the kinetochore microtubule coming from opposite poles. Okay. Alright, so next is the anaphase. So you can see that the cell is a bit longer compared to the prophase, prometaphase, and metaphase. So centromere of each chromosomes break apart during anaphase. So the protein of centromere that brings the sister chromatid together will break apart. So the chromatid now can move to the opposite poles of the cell. The movement of chromatids is due to the shorten of mitotic spindle or the kinetochore microtubule. Let's see how kinetochore microtubules shorten during anaphase. So this one is the experiment. You can see that here is the marking point whereby the kinetochore microtubules will be shortened. So the result is the shorten of the mitotic spindle. So in anaphase, sister chromatids separate and move along the kinetochore microtubule towards opposite ends of the cell. So the microtubules shorten by depolymerizing at their kinetochore ends. Okay, next is the telophase and cytokinesis. So we first take a look on the telophase first. So both sets of chromosome reach or re-reach at opposite poles of the cell. So now the chromosome begins to become less condensed. Now it becomes a thread of chromatin again. No more centromere, right? It becomes single chromosome again, and there is no mitotic spindle. The spindle microtubules depolymerize at all, and then the nuclear envelope and nucleolus reappear. Whereby the cytokinesis, you can see here that the cell already forming the cleavage furrow. So this one is the animal cell because there is a formation of cleavage furrow. So cytokinesis means the division of cytoplasm. So in animal cells, cytokinesis occur by a process non-cleavage, formation of the cleavage furrow, while in plant cell, the cytokinesis form a cell plate because plant contain a cell wall. Alright, so this is the difference between cleavage furrow in animal cell and cell plate in a plant cell. Alright, this one is the example of mitosis in onion root cell. You can see here is the prophase, whereby it still have the nucleolus. You can see that the chromatin, the chromosome not yet condensed but already condensing in prophase. Okay, and then the here is the prometaphase. And then metaphase is see all the chromosomes line up in the metaphase plate, and then during anaphase, each chromosome will pull toward the opposite pole by the mitotic spindle because mitotic spindle depolymerize. And then telophase, you can see here is all chromosomes already reach at the opposite cells, and then there is a formation of a cell plate because this is a plant cell, onion. Alright. There is some differences of mitosis in animal and plant cell, whereby in animal cells they have centrioles and ester, but in plant cell they have no centriole and ester, but they still have the microtubule or mitotic spindle. And then in animal cell, the cytokinesis involve the formation of cleavage furrow, while plant cell, the cytokinesis involved in the formation of cell plate. Animal cell mitosis occur in somatic cell, while plant cell, the mitosis occurs in the meristematic cell.